Hello, everyone, and welcome to today's webinar, An Epic Advance in Epigenomics. Automated cut and run enables chromatin profiling for scaled research and drug discovery applications. I am Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit www.beckman.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I do also want to mention that there is a survey at the end of the webinar. If you could please take a moment and submit your responses when prompted to do so, that would be greatly appreciated. I'd like to now welcome our speakers. Andrea Johnstone, Senior Director of Product Development at EpiCypher, and Laura Euler, Senior Manager, Drug Discovery Genomic Solutions at Beckman Coulter Life Sciences. Lori, you may now begin your portion of the presentation.
Great. Thank you so much, Cassie, and thank you everybody for joining us today. We appreciate you taking some time to learn a little bit more about this technology. I'm going to jump in with a little bit of, some basics. So let's start at the beginning. Let's start at the basics of nucleic acid purification. The way we tend to look at things at Beckman is there's really two paths for nucleic acid purification, and it all really depends on your starting material. We know that our ultimate goal is purified nucleic acid, so that's great. But we can either start with biological materials where we have a lysate, very dirty sample, or we can start with something like an in vitro reaction, so an enzymatic reaction, something like that. So these are the two pathways that can generally happen as we look to do nucleic acid purification. There are two main ways that these purifications can happen. One is a chemical route. So I'm sure most people are familiar with kind of the old school phenol chloroform. A lot of challenges with this type of method. The main one being using chemicals that really are not not healthy for the researchers to be using. So not the most preferable way that folks would like to do nucleic acid purification. Most of us are really familiar with solid phase. So they're, obviously silica membrane. So those are column or those are plate-based methods. And these are all based on silica gels. So DNA will bind under certain situations. It's all buffer dependent. And the other option are mag beads. So mag beads can bind to the surface. The nucleic acid will bind to the surface of these beads, and then it's just a simple wash. And then removal of the nucleic acid from the bead. So I'm going to talk to you a little bit today about SPRY-based technology. And SPRY stands for solid phase reverse immobilization. And what this means is that under one set of conditions, your DNA is going to bind to the beads. You simply do some washes to purify your sample, and then you change the environment and the DNA will no longer bind. So really simple process of bind, wash, and then elute. On the left-hand side of this slide, you can see that we have selective binding of nucleic acid. Only nucleic acids are going to bind or be immobilized onto the surface of the bead. Again, this is what allows you to do the washing step that gets rid of everything else that might be in the sample. So if it was your crude sample, you're going to get rid of all of that cellular debris that's in there. If it was an enzymatic reaction or an in vitro sample, you'll get rid of anything else that may have been in there, any of the enzymes, buffers, etc. So then you're going to need to release the nucleic acid from the bead. You can alter the conditions, and that's what releases the nucleic acid from the bead. The purity of your sample is really dependent on that selective binding onto the bead. So the selective binding of the nucleic acid on the bead, that's what controls the purity. The yield itself is dependent on both the binding and the ability to release that DNA from the bead. I'm going to give you a little bit of a brief history of SPRY technology. So this was originally developed in the 1990s. It was developed at the Whitehead Institute at MIT. And the whole purpose of doing a technology like this was as part of the Human Genome Project. And it was developed to make PCR cleanup easier for Sanger sequencing. And at the time, that was the only method to do the sequencing for the Human Genome Project. In 2000, Agincourt Biosciences was established with the goal being to commercialize this technology. And then in 2005, Beckman Coulter acquired Agincourt Biosciences. And so spray technology is now the hallmark of the genomics portfolio for Beckman Life Sciences. So I'm going to talk a little bit about these reagents, how it actually works, and why it's a preferable way to do your nucleic acid isolation, or either your extractions or your cleanups. So there's three main components. The first is the magnetic bead itself. So that bead provides that solid support for the binding. So you need something that this whole reaction, this binding is going to happen on. So that's your solid support. You then have a crowding agent, and the goal of the crowding agent is really to basically squeeze your nucleic acid out of the solution and drive it to bind onto the beads. The concentration of that crowding agent is really critical. So that concentration is really what determines that size selection. So that's what's going to, that crowding agent determines the size of the nucleic acid that's going to bind to your solid surface magnetic bead. The last component here is the binding buffer. So these binding buffers contain salt ions, and you can adjust the pH for binding. The pH really has a huge impact on the charge here. And so if you have a change in the pH, that can decrease your binding efficiency. So it's really important, if you're going to use a system like a spray-based reagent, that you're considering the buffer that your sample is in to begin with. Because again, pH plays a huge role in how this happens. So let's talk a little bit about the beads themselves, because they're really the magic in this whole process. There are a few really critical parts here of these beads, and you can see this is kind of a, if we took a bead and we cut it in half and we looked at it, this is what it would look like. This surface layer is coated, and this engages in the binding of the nucleic acids. This is really the hallmark to low non-specific binding, this coating of the membrane. So you're not going to get things other than your nucleic acid binding onto the surface of this bead. The next layer that we see as we go in from that carboxy-coated surface is our magnetic layer. This is really important. This is what allows the manipulation of the beads during the workflow. Having a magnetic bead is a really interesting technology. It's a really interesting way to move things around, and it's quite easy. So this is one of the features that allows this technology to be easily automated. The last thing we're going to look at is the center. So this is a polystyrene core, and the purpose of that is for buoyancy. You don't want these beads to just sink to the bottom. If they did, there really wouldn't be an optimum interaction time with the nucleic acids. So having that buoyancy allows them to stay in solution and gives them longer time to actually interact with the nucleic acid that you're trying to bind. If you look on the right-hand side, you're going to see a picture of the actual surface of these beads. We always say it looks a little bit like cauliflower. That surface topography is very important. Because of that topography, you have a high surface area, and that's really what promotes that high binding of nucleic acid. So more surface area, more nucleic acids that can bind. The other thing that's really important is the optimization of the size of the bead itself. So the bead is within a really tight size range. This is really done both for the recovery and the manipulation of the beads. So as I mentioned earlier, this is a really simple process. One of the things that a lot of customers talk to us about is how they're very familiar with column-based technologies. So either a spin column or a 96-well plate with a silicon membrane in there. That's great. They are simple processes, but folks who want to scale up, folks who want to do an automated process, a lot of times, look to that because 96-well plates are something very common for them. This method is really easily automatable because it's just 3 simple steps. So again, we're going to bind in the presence of a proprietary buffer. That's where our nucleic acids are going to bind to the surface of our beads. We're going to immobilize these beads onto a magnet. So you can see in the picture in the middle, there's just a little ring magnet that goes around this tube. It's going to pull the beads that are bound to the nucleic acid over to the sides. This allows you to wash out anything that's in that sample that we don't want. So anything that is not bound to those beads, so it's not nucleic acid, can be washed out at this step. And then the final step, which is probably the most important, is that elution step. So again, you're going to add a buffer that changes the environment that these beads are in. And at that stage, the DNA is released from the beads. The beads are still immobilized on the side, so they're still attracted to that magnet. So at this stage, it's really easy to just simply pipette away your nucleic acid or your sample of interest. And again, this process is easily automated. So I'm sure a lot of you are familiar with a lot of the technology that Beckman itself has, our Biomech systems. We have protocols that we've worked out for our kits on our Biomech. So it's really a kind of plug and play solution for customers. This just shows you exactly what this looks like. I think if you've not used magnetic bead-based technology before, it's a little bit hard to conceptualize. So basically your magnet's on the bottom. When it looks a little bit dark, that's when you add the beads. And then you see that, this is a time-lapse video, but you see that quick pull down of the beads down to the magnet. This is kind of a top-down view of that. Again, a lot of folks have concerns about being able to introduce a new technology such as this in their lab. This is just to illustrate to you how easy it really is to work with these beads. So as they're pulled to the side of the tube, it's quite simple to go in and out of the middle of that tube to remove sample, to do your washes, things like that. So again, really easily automatable process. There's a bunch of different magnets that can actually be used, again, because these are automation friendly, but these sorts of plates can also be used if you're doing a manual process. So some people maybe aren't doing high throughput and they want to be able to perform these steps just on the bench. So there are magnets that are automation friendly, and there are magnets that you can use just in a manual process on your bench. There are two key types of magnets, or two primary types of magnets. One is the ring, and that's what the earlier picture showed. If you look on the top left here, though, this is a bar magnet. Essentially, the only difference is that pulls the sample all to one side, whereas the ring magnet pulls it in a circular fashion to the outside. So again, when you look at these kits, there's very simple things in here, things that everybody are familiar with. There's a lysis step, so there are lysis buffers. You're going to have a nucleic acid digestion step in there. So you'll digest the type of nucleic acid that you're not interested in. You can have some rebinding buffers that aid in your washing steps and post-wash. And the goal of all of this is for the removal of those contaminants. So either contaminants that were introduced with buffers you used earlier in the process, or that are very specific to the sample type that you have. And I'm going to kind of summarize a little bit what's unique about spry bead-based technology and why it's really great to use it in things like the cut-and-run process. They're paramagnetic particles, easily automated. Again, it's really ideal for high throughput labs. You don't need centrifugation. Any sort of high throughput technique where you end up needing to pull off and add in a centrifugation step still has a lot of manual manipulation. So no centrifuge or filtration needed. Again, that uniform bead size really gives highly reproducible results. That negatively charged surface, it allows you to have all of this occur in an aqueous solution environment. So that's really great and gentle on the samples that you're looking to do downstream work with. That non-polystyrene polymer surface, again, that load 9 specific binding is really important in these processes. And then that high surface to mass ratio, again, extremely important to have that increased binding capacity. There's a very fast magnetic response, as we saw in that time-lapse video, so you get very rapid purifications. These size optimized particles have a very low settling rate, and again, makes it ideal for automated applications. Just very briefly, I want to let you know that in addition to things for cleanup, which we'll be talking about a little bit later in this webinar, Beckman also does offer solutions for sample preparation, so for DNA extraction solutions. One of the really great things is you can go on our website and you can look up what would be the best kit for your application. based on your input material, you can look up based on your output, or you can look at your downstream applications. It'll help you pick what kit would be best for your particular application. We also have kits that are for DNA and RNA extraction, so for total extraction, for RNA itself, and as I mentioned, for bead-based cleanup and size selection reagents. And I just want to remind you that we will have a Q&A session at the end, so please feel free to put your, please feel free to put your questions in that Q&A box. But right now, I'd like to turn it over to Andrea Johnstone.
Awesome. Thanks, Lori. That was a great lead into what I'm going to talk about. And so I'll just jump right in here with the take-home messages. So first, if you want to understand how genes are regulated, we really need robust technologies that are capable of mapping distinct features of chromatin. And looking at DNA methylation and chromatin accessibility get us part of the way there, but there's enormous additional diversity at the level of chromatin-bound proteins and modified histones. And the most common method that people use to study this are CHIP, at least historically. But in recent years, a new technology called CUT&RUN has really been supplanting CHIP as the go-to epigenomic mapping assay. And then finally, the meat and potatoes of what I want to talk about is how EpiCypher has leveraged some of our proprietary technologies and unique expertise to automate CUT&RUN. And this is certainly enabled in large part, as Lori talked about, by scribeads. Okay, so for anyone who might not be familiar with EpiCypher, you can probably gather by now that we are a biotechnology company that's focused on innovating new technologies to advance our understanding of chromatin and gene regulation. And during this talk, I'll focus more on our ultrasensitive epigenomic mapping technologies and how these are enabling us to better understand not just basic gene expression changes during normal development and how these processes go awry in disease, but also how these technologies are integrating with this rapidly expanding field of therapeutic development programs that are trying to manipulate gene expression, either through small molecule inhibitors of epigenetic enzymes, transcriptional regulators, or even cell and gene therapies. We are trying to fundamentally reprogram a cell's gene expression states. And also I'll talk about how these technologies can be integrated to discover novel biomarkers that previously we were unable to access because we were focused on other aspects of chromatin, or focused on genetics, for example, which fall short of identifying, biological changes that are sensitive to the environment in some cases. Okay, so the most commonly used epigenomic mapping approaches in the field are ATAC-seq and bisulfite-seq, which look at chromatin accessibility and DNA methylation respectively. And as I said, these give us great information about chromatin regulation, but they give us a largely binary view of chromatin. And biology is not always that black and white. So for example, you can have open or accessible chromatin where genes are not being actively transcribed. So there's either some other factor here that's blocking transcription, or there's something that's missing that's not, promoting active transcription. And in the case of bisulfite sequencing, looking at DNA methylation, this is commonly associated with repressed regions of chromatin. However, you can actually find a large amount of methylated DNA active gene bodies. So again, it's not as black and white as we would want it, as it would seem to be. And so if we want to drill farther down and get a higher resolution view of how chromatin is regulated, ChIP-seq historically has been a very common approach to map the specific localization of transcription factors that are involved in certain developmental or regulatory processes of interest, and also to look at modified, various flavors of modified histones that demark specific regulatory elements on chromatin, like enhancers, promoters, active gene bodies, and either dynamically or stably repressed chromatin. And so ChIP-seq depends really on the specificity of your antibody. And this is particularly problematic when it comes to histone PTMs, because asking an antibody to distinguish between a mono versus a di versus a trimethylation at one specific site is quite a tall order. And in addition, antibodies might cross-react across lysine sites. So an antibody that binds K4 tri, if it cross-reacts with K9 tri, for example, this can really confound your biological interpretations. And EpiCypher is really well positioned to address this issue because one of our core competencies is developing fully defined semi-synthetic nucleosomes that are used in a variety of different types of research applications. We have over 100 of these available in our catalog. And a couple of tricks we can do is we can engineer those histone PTMs that I just talked about on the last slide. We can site-specifically engineer them on the histone tails. And then we can take those unique PTM nucleosomes and wrap them with unique sequences of DNA, a so-called DNA barcode. And this enables us to assemble these nucleosomes into rational panel sets that comprise both the target that you're chasing in a ChIP-seq experiment, as well as highly related off-targets. So I'm showing here our lysine methylation, or K-MET-STAT panel, which has mono, di, and trimethylated nucleosomes, each uniquely DNA barcoded at distinct sites. So K4, K9, K27, and so on and so on. And so what we do is we take this panel and we spike it into a ChIP experiment, and we ask a simple question by looking at the barcoded DNAs that you recovered at the end. And you say, was my antibody specific, and did the assay work? And so we tackled these questions in earnest by doing a large-scale study of almost 400 commercially available antibodies to histone PTMs, lysine methylation and acylation states. And what we found, sadly, is that about 70% of the antibodies failed. Either they weren't specific or they were not efficiently recovering their target. They failed to give you a lot of DNA yield at the end. And unfortunately, the case is no better when you look at the antibodies that most people in the field use. So we had tested 24 of the top cited PTM antibodies for just highly studied PTMs, comprising almost 5,000 different citations. And so these are the antibodies that really everybody uses over and over. And unfortunately, the failure rate for these approaches 80%. Interestingly, all of these antibodies were very high efficiency. They were very good at recovering DNA. Unfortunately, that DNA was not specific for the target that you think that you were mapping. And so this can obviously confound your biological interpretation. So if you're interested to look at this data, it's all publicly available on chromatinantibodies.com. You can either search your antibody of interest and observe its specificity in a ChIP-seq experiment, or you can search whatever PTM you're interested in studying and get our recommendations for the best antibody. Okay, but along this path, we realized, what is our goal here? ChIP has other problems that even rigorously defined controls can't fix. It requires millions of cells, so it's inaccessible to primary cells and precious clinical tissue. It's laborious, you have to optimize it, and it's very low throughput. And the data quality is not great, even when you're spending a lot of money to sequence, to resolve your signal from your noise with high sequencing depth. And so a few years ago, we actually recognized very on the value of this new technology termed CUT&RUN, developed by Steve Hennickhoff's lab at the Hutch. And we were actually first to market with this technology that enables you to map genome-wide the localization of histone modifications and transcription factors using 100-fold fewer cells than ChIP, using a very robust and user-friendly workflow, and get data at the end of the day that has very high signal-to-noise, even with 10-fold reduced sequencing depth. So reaping massive benefits in cost savings and just overall labor and effort. And the nice thing about this workflow is it was very amenable to those very similar flavor of those nucleosome spiking controls that I just talked about for ChIP. And so we had to go back to the drawing board and redesign our spike-ins for CUT&RUN because the workflow is a little bit different. So in ChIP, if you remember, you're fragmenting your chromatin and then you're using immunoprecipitation to try to fish your antibody out of solution and wash away everything that didn't bind. In CUT&RUN instead, you're taking your intact cells, so there's no heavy sample processing here, just cells that you immobilize to a solid support. So we took our spike-in PTM-defined DNA barcoded nucleosomes, and we immobilized them to a solid support as well, just like your samples are immobilized. You then add an antibody to your target of interest, and you diffuse in this protein AG immunotethered construct. So the protein AG will go and bind wherever your antibody is bound on chromatin and in your spike-ins. and then you activate the enzyme that the protein AG is conjugated to, which in this case is micrococcal nuclease. And that will cause your target to diffuse into solution, both from the sample chromatin and from your spike-ins. And so if you isolate the DNA associated with your protein of interest, now a small portion of your sequencing libraries have the barcoded DNA from your spike-ins, and you can then look at recovery of your on-target histone PTM compared to all those highly related off-targets, and ask a simple question: did my assay work? Was my MNase cleavage optimized? Were my wash conditions good? And is my antibody specific for the target of interest? And then downstream, you can do quantitative comparisons using this as a normalization standard. Okay, so instead of doing a large field survey like we did in CHIP, we said let's get right to the heart of it and find the best in class antibodies that we can find for cut and run. And so the heat map on the left is showing you the best antibody we found for 15 different lysine, widely studied lysine methyl states. Each row is an antibody, and each column is whichever spike in nucleosome they recover. So a specific antibody will show you a blue box for the target nucleosome surrounded by red. So you can see one row down here, if I can draw real quick. So this row right here is showing you an antibody that is not very specific for its target. So we're still trying to look for antibodies to that mark. But for every other mark, we've identified a very highly specific antibody that can be used to uncover the unique biology associated with each of these modifications. But specificity isn't the whole game. We also want antibodies that are high efficiency. Remember that one of the advantages of cut and run is you can use 100-fold fewer cells than you can to CHIP, so down to just a few thousand cells. But the caveat here is that not every antibody can do this. You really need antibodies that can have high efficiency for their target that can enable you to reduce cell numbers. So how we look at this is we simply do a cut and run experiment at a standard amount of cells, 500,000, and then 10-fold reduced cells, 50,000. And so you can see the antibody on the left. Oh, and what we're showing here is called peaks. So looking at when we did cut and run with this antibody, where is that protein enriched on the genome, a so-called peak. And so the Venn diagram represents the number of peaks we called at the standard cell input in orange, and then at 10-fold reduced cell inputs in blue. And what you can see for the H3K9 acetyl antibody here is that you've lost most of your peaks, making that jump down to 50,000 cells. However, the K4 monomethyl antibody here on the right retains over 84% of its peaks, even at 10-fold reduced cells. So this is a very high efficiency antibody. So now that we have those defined spike-in controls that enable you to confirm your assay works, we have very robust positive and negative control antibodies. We set out to develop user-friendly, streamlined kits to enable people to do cut and run in labs all over the world. And we started out trying to streamline the protocol, and I'm glad Lori gave an introduction to PCIA-based DNA extraction, because this is the first generation protocol that was developed by Skeen and Hinnekoff to isolate DNA at the end of cut and run. And as Lori mentioned, it's a very robust historical approach, but it's not the most user-friendly. So for the first iteration of our cut and run kits, we started using DNA purification spin columns because we thought, this is pretty easy for most people to do. And one thing that we realized right off the bat, so the graph on the right here is a graph of cut and run DNA yield. So everything in the assay was the same, except for which vendor we got a DNA purification spin column to isolate the DNA. And what you can see here is a dramatic difference in the recovery of DNA across different spin column providers. So the one we selected was the best, and that's in orange. And this is really important because the amount of DNA that you recover after cut and run and the size, the fragment distribution of that DNA are the two best indicators of success that cut and run worked, and that you can prepare libraries and move on to sequencing. And so it was really important that we got the best DNA purification protocol that we could get at the time. All right, so based on this, we developed not only a cut and run kit, but also a downstream library prep kit that is specifically tailored to prep NGS libraries from the very low inputs you get from cut and run DNA. And this is these kits are very robust for a variety of targets. I spent a lot of time talking about histone PTMs, but they also work very well for chromatin-bound proteins like transcription factors, epigenetic modifying enzymes, and even chromatin remodeling enzymes, which are very difficult to profile in ChIP-seq. And we're very proud of these kits. They've been adopted in over 2,000 labs globally. However, one thing always bothered us about the workflow of these kits. So this is what it looks like. So you start out preparing cells. You're usually batch processing your cells unless you have very unique sample conditions. And then you distribute those cells into 8 strip tubes, and you move through the steps of cut and run using a multi-channel pipetter, which enables you to process multiple reactions simultaneously, and your reproducibility across replicates is extremely high, and overall the workflow is just seamless and streamlined. However, then you go to your old school single channel pipette, you distribute your cut and run DNA to individual spin columns, and then you have to, one by one, sequentially process your samples through the DNA purification steps. And then for library prep, you're thankfully back to your multi-channel pipettes and parallel processing here to make your final NGS libraries. Okay, so obviously this workflow is not ideal, and a really easy solution for this was right in front of us all along, and Lori did an excellent job introducing why, and that is using spry beads to isolate your DNA, because those are, as she mentioned, compatible with multi-channel pipetting and automation. And so we wanted to make sure that if we're swapping out this really critical aspect of our cut and run kits, remember I showed you a few slides ago that the method you use to recover DNA, even different spin columns, make a big difference in the success of your assay. So we wanted to make sure if we're swapping from our gold standard spin columns to spry beads, that the performance was just as good, if not better. And so here on the left, what you see is a comparison of DNA yields recovered using spin columns in orange and various ratios of spry beads, because we really want to make sure we're tuning the ratio of spry to maximize the amount of yield we get and also target the specific fragment sizes that you want to target from cut and run, which is a mononucleosome. And we did this by titrating down the number of cells that we input into cut and run, because we wanted to make sure that going from spin columns to spry beads, we didn't lose any sensitivity to drive towards those low cell numbers. And what you can see, at 500K and 100K cells, things look pretty similar. The more spry beads you add, maybe you lose some yield, and same with the less. But once you get down to 50K and 10K cells, the yields are actually better in spry beads. And if you look on the right here, so these are tape stations, so this is showing a size distribution of what are the sizes, the fragments of DNA that you recovered in cut and run. And we're just comparing spin columns on the left to spry beads on the right. And here I will draw again, because I will draw your attention to this area here, where when you titrate down cells, you actually get better recoveries, better size distributions with the spry beads at those low cell numbers compared to spin columns. So that was great to see. And so we did a series of follow-up assays, alpha testing, all sorts of things to make sure that this protocol change was really, really robust for our kit users. And I'm proud to say that our new version, updated version of these kits is going to be coming out literally within the next 2 weeks. So this is perfect timing to talk about this. And so now our kits, are incredibly streamlined, going from cells all the way through to NGS libraries using multi-channel pipetting. All right, so what we realized about this time is, we've kind of put together all the pieces of the puzzle that we need to take cut and run from the bench, where you're doing multi-channel pipetting. Yes, it's streamlined, but it's still, limited in terms of scale. And so now we can take that and actually automate it and scale it in a way that's never really been done before. So we had a robust assay. Our protocol is incredibly reproducible. We had those defined controls, those nucleosome spiking controls that I told you about. We have validated antibodies that we know work great in cut and run. And now with the inclusion of spry beads in our workflow, we have a truly scalable workflow. And this is really a major advance, because if you think about, why didn't people do this before? It's because ChIP is incredibly hard to automate. Some people have done it, but it presents lots of problems. So for example, In CHIP, you have to fragment and most frequently sonicate your chromatin in order to input it into the IP reaction. And that has to be optimized for different cell types and sample types. However, in cut-and-run, your input is intact cells or nuclei. So it's a pretty standardized process to just put these cells in and then move it through the SOP of cut-and-run. Also, in CHIP, you needed millions of cells. And so any sample that you tried to automate just wouldn't get you that far. You wouldn't be able to profile that many features. But with cut-and-run, we can get down to as little as 5K cells. So any precious samples, especially clinical samples and primary cells, can get you more information. Another important thing is sequencing depth. So in ChIP-seq, you needed 30 million reads, cut-and-run 10-fold less reads, which enables you to multiplex all of those reactions onto a single sequencing run and get data faster and cheaper, by the way. So about 70% cost savings in cut-and-run compared to CHIP. And then again, your data quality is actually better in cut-and-run, and your experimental throughput is high just because the workflow in cut-and-run is compatible with 96-well plates. And so around this time, the impetus also for us to try to automate cut-and-run came from a connection we had with the Immunological Genome, or InGen Consortium. So this is an international consortium led by Christophe Benoit and Diane Mathis to really chart the mouse innate and adaptive immune systems. This is a very large-scale effort bringing together dozens of labs all across the world to collect unique types of mouse immune cells at various stages through differentiation, various pathogenic challenges, and genetic backgrounds in order to better understand immune diversity and immune responses. And they had been trying to figure out how can they map chromatin features. They had dabbled with the idea of doing CHIP, but the problem with CHIP is that they could only guarantee that they could get 10,000 cells per sample. And this was just a non-starter for CHIP. And so the reason they were only getting 10,000 cells per sample is because they were taking mouse, isolating primary cells, and then FACS sorting them for different immune cell markers to get really unique populations of immune cells that they then wanted to profile not just one, but multiple features, unique regulatory elements of chromatin using just one population of cells. And so they challenged us to basically optimize cut-and-run for this system using just 10,000 cells per individual feature that we were able to mark. Okay, so this is the process that we set about trying to automate cut-and-run. We did all the standard things, making sure our key controls were incorporated, optimizing our buffers to make sure DNA yields were optimized and stickiness wasn't a problem for the robots. Of course, we had spry beads in there for the maximal throughput and consistency. And then at the end of the day, we made sure the assay was very reproducible and sensitive. We didn't stop at just cut-and-run. We also automated lots of the quality control checks. So using a fluorescent-based DNA quantification system to look at DNA yields. And then we miniaturized our library prep protocol on the robot as well as our tape station prep to look at size distribution of the final fragments and normalize and pool the DNA for sequencing. And again, just making sure that these steps were reproducible and sensitive. And then the final step was really to tailor this process to the MGen specific program needs. And so one challenge here was optimizing preparation of the samples. And we had previously known that sometimes, in most cases, using native cells works very well in cut-and-run. But in some cases, like lysine acetyl marks, these are rapidly turned over by HDACs, which remove these marks from chromatin. And so what we found is that using a light fix, not a standard chip fix, just 0.1% formaldehyde for 1 minute, was enough to stabilize that K27 acetyl mark. So now you can see at the bottom track here, which I will draw on, you can see this track here shows much clearer peaks as opposed to this top track where we didn't fix. So now we have sample processing conditions that are optimized for the targets of interest. We also needed to optimize nuclei preparation and enable all these labs from all over the world to ship nuclei to us in very good condition to do the cut and run. And we found an optimal nuclei buffer to do that, to really stabilize these nuclei for shipping and for cut and run. And we actually just launched this buffer last week. So it can isolate nuclei from a variety of cell types and tissue. And then the final step here was to identify good antibodies that can profile all these diverse regulatory marks, not only being specific, but also being efficient, being able to drive to low cells. And I'll note here that some of these marks, we had really high failure rates. So H3K36 trimethylation, we tested 10 antibodies. These aren't 10 random antibodies. These are 10 antibodies that looked great, bound to nucleosomes and some of our surrogate assays. So these are the best, the cream of the crop already, and only 2 of these out of the 10 tested passed. Most of them were not able to get good signal down to just 10,000 cells. All right, so that was our optimization workflow. So at the end of the day, what we were able to do is develop a fully automated cut and run assay that reduced our hands-on time by half and increased our overall throughput, or our total number of reactions per week, by over 8x, just for a single robot. Okay, and here's a look at some of the data coming off of auto cut and run for 3 different marks. H3K4 trimethyl, a mark of active promoters. You can see these nice sharp peaks here. And noticeably, the peaks are retained, going from 500,000 cells all the way down to just 5,000 cells. By the way, this is optimization in K562 cells. We haven't started with immune cells yet, primary immune cells. And you can see this trend was reproduced across different targets. So K27 tri, again, this broad marker of intergenic and repressed regions is retained when you go down to 5,000 cells. And then K36 tri, a very specific marker of active gene bodies. You can see that peak structure is also maintained down to 5,000 cells. On the right here is a map of Pearson correlations going on those different cell titrations. And what you can see is that the results are very reproducible and robust, going all the way down to 5,000 cells. So now we have a robust system. We also wanted to make sure that the system was protected from potential error. When you're running things in a robot and something goes wrong, you're not a human that's watching it by eye and can maybe flag if a pipette error occurs. And so that's where our spiking controls really proved to be useful, and monitoring the specificity of our antibodies and overall reaction success. And so what I'm showing on the left here are 4 different samples. So these were provided from 4 different MGen labs, and we ran them all on the same auto cut and run plate using the same antibodies. So the data should look similar, right? The antibodies should be specific throughout. You can see this one sample down here, and for some reason the drawing's not working. There you go. And it also gave a lot of high background here in both targets, H3K4 tri and K27 tri. And so we flagged this as being potentially problematic because it had low signal to noise compared to other samples tested using the same antibodies. And the same thing happened in the genomic data. So this just didn't have great recovery in the genomic data. If we only had this information here on the right, we might not be so confident to throw this out. We don't really know what happened here. Is that really, a reflection of the sample or the genetic perturbation or the treatment? We don't know unless we have those defined controls that really show that there was a problem with that overall sample preparation. Okay, so now that we have robust antibodies, a reproducible workflow, and everything's protected by those defined controls, now we can start getting to work and mapping those 10,000 FACS-sorted cells. And so what I'm showing you here is type 3 innate lymphoid cells, and I'm just showing you the diversity of features that we were able to map with just 10,000 cells per reaction. So you can see K4 mono, a mark of poised enhancers, contrasting with K27 acetyl, a mark of active enhancers. You can see K4 trimethyl, a mark of promoters. K27 tri shows a totally unique distribution because it's associated with repressed genes. K36 tri is a more broad mark of active gene bodies. You can see it kind of, in between the promoters here. And then CTCF, which is a transcription factor that's typically bound to enhancers as well. So we were able to mark very diverse features with low numbers of starting cells and with high signal to noise. And so we're still in final data analysis right now. So hopefully over the next few months, we should be talking more about what our overall findings here. But excitingly, we've already started to do what we call epigenomic fingerprinting. We've already started to see signatures that are associated with distinct cell types. So on the top is our IgG negative control. It's low across all cells, as you would expect. But in the middle here is K4 tri, a mark of active promoters. And what you can see is that K4 tri at this SERPIN6b locus is completely absent in granulocytes. However, it's present in the other two types of immune cells here. And the converse is true when you look at K27 tri, a mark of repressed genes. So in granulocytes, K4 tri is low, K27 tri is high, which might mean that this locus is inactivated. And then you see a completely different pattern at the next locus, which I can't pronounce, PXYLP1, where in granulocytes K4 tri is high, but then K27 tri is absent, and this is distinct from the other two cells. So this is very exciting to see, and we're looking forward to seeing how the data comes out at the end. Okay, so switching gears now. So I told you about how automated cut and run can be used to enable consortium scale genomics, and in a specific application where you want to fingerprint, you want to find unique signatures associated with different cells. But another potential application of auto cut and run is looking at biomarker discovery. So clinicians have freezers full of patient samples, and they really want to use these to look at, are there specific features of chromatin that might enable us to better understand how genes are regulating disease, and maybe even subclassifying patients. Are there patients with unique chromatin signatures that might make them poised or prone to have different responses to drugs? And so in order to do this, we need to be able to access those oftentimes flash frozen samples. And so we collaborated with Shahzad Sheikh from UNC to try to optimize protocols to do just that, because he's a clinician who treats patients with Crohn's disease, and he had freezers full of flash frozen intestinal biopsies. So what we were able to do is optimize a protocol to extract nuclei from these flash frozen cells. And when we looked at their at cut and run data, we compared Crohn's disease from healthy controls, and we compared that to freshly prepared cells for a variety of different marks, some histone PTMs and transcription factors. And we were able to optimize a protocol that shows similar profiles, good signal to noise between frozen tissue and freshly prepared cells. So this is really exciting to see, and we're now working on scaling this even further and starting to put patient samples through automated cut and run to see if we can find new biomarkers. And another exciting aspect of this is that, if we isolate nuclei from that frozen tissue, these are genome-wide heat maps looking at the enrichment in red compared to background in blue at transcription start sites. So you see K4 tri is very heavily activated at transcription start sites, and K27 tri shows this oppositional enrichment pattern, which I'll sort of circle here. And what we're able to do is that even if we isolate nuclei at the site of the clinician and then cryopreserve it, we see that the genomic profiles are largely the same. And this enables multi-center collaborations because people can prep their nuclei in their own labs and then ship it to one common site to do the cut and run, and you don't lose much in terms of data quality. All right, and lastly, another obvious application of automated cut and run is reading out drug response. So if you're trying to inhibit a transcription factor or an epigenetic enzyme, can you use this in vivo cut and run assay to look at localization of your target onto chromatin? And so this slide is showing proof of principle that yes, we can do this. So in this experiment, we looked at treatment with this Syndax inhibitor, which is an inhibitor of menin. So menin is involved in the pathology of leukemias that are known to have genomic translocations of menin and MLL proteins. And if you inhibit, menin, it can help in the outcomes of these patients. And so what we did here is we mixed cells either treated with this inhibitor or the vehicle control at discrete quintile ratios. So these cells are, so these samples either had 20, 40, 60, 80, or 100% treated compared to controls, because we really wanted to see, you know, does this assay show good sensitivity and good linearity when you're inhibiting the target. And so if you just look at the tracks at the bottom here, what you can see is when you add more and more menin-inhibited cells, you can see localization of menin is depleted from target chromatin in a very linear and quantitative way. And then there's some locations where you can also see K4 tri, which is a downstream epigenetic mark catalyzed by menin in the MLL complex, and you can see that going down as well, while other marks like K27 tri, which are not involved in this pathway, are unchanged. And you can use, you know, large-scale differential occupancy analyses to identify genes that are up and down regulated in response to treatment. So overall, this shows that cut-and-run can detect quantitative changes in drug-induced enrichments. All right, so then just to wrap us up here and take a step back. So as we set out to deploy auto cut-and-run for some of these advanced applications, we're really, really well positioned to do this just based on our overall experience with cut-and-run, where we've worked with dozens of partners, both academic, biotech, and pharma collaborators to use cut-and-run to help answer their research questions. We've demonstrated using literally hundreds of distinct sample types that cut-and-run can be used for both primary, sorted, drug-treated cells, as well as tissue. I showed you some data at the beginning about all the antibodies we screened, and so we're very serious about finding good antibodies. I focused mostly on histone PTMs, but we've actually tested thousands of antibodies in total and found great antibodies for PTMs as well as chromatin proteins like transcription factors or chromatin modifiers. We can handle very large projects. So I told you about the MGen consortium that's doing cell identity fingerprinting. And in total, we ran over 2,000 reactions for that consortia. But in total, we've run over 10,000 cut-and-run reactions. So we have a ton of experience. We know how to optimize conditions for different experimental variables and how to flag things and troubleshoot if things go wrong. So at the end of the day, when you put all these sort of stats together, really the amount of new things we can do with this technology, the new applications that we can access that were previously inaccessible by chip is limitless. So as you leave the webinar, I want to challenge you today to ask how cut-and-run and even auto cut-and-run can accelerate your research. So if you want to get started, I told you about our cut-and-run and our library prep kits. We have a new version coming out in just a few weeks using SPRI-based DNA purification, so that'll enable really streamlined processing. But if you want to do auto cut-and-run, you know, I showed you some vignettes about, you know, how we can do pilot studies just to optimize conditions all the way up to high throughput projects. Importantly, we're not stopping at cut-and-run, so we are also automating other modalities like DNA methylation, chromatin accessibility, and multi-omics as well. So our experts can help you set up cut-and-run in-house, or very exciting, we are about to launch cut-and-run services actually in November, so next month. We've been running these services sort of quietly behind the scenes, and it's been very popular, so we haven't had the bandwidth, frankly, to launch, but we're now very excited to announce the launch next month. So if you're interested in setting up a fully customized project to suit your research needs, please contact us at services@epicypher.com. We'll also be sharing a poll. Would you like to be contacted by an EpiCypher representative? So you can say yes to that question as well, and we will reach out. And then lastly, I just want to thank Beckman for the opportunity to do this webinar. I think, you know, the way Lori and my presentations integrated was just really awesome. And we thank you guys for the opportunity, and to LabRoots as well for hosting.
Thank you, Andrea and Lori, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's go ahead and get started. Our first question says, what liquid handling instrument do you use for automation? Could the Biomech i7 be used?
Absolutely, the Biomech can be used. So the beauty of this platform is it's really agnostic to any instrument. It should work on anything. And in fact, the first iteration of automating cut and run was done by Steve Henikoff, where he used a Biomech. So yeah, if you want support setting up automation in-house, please reach out to us and our experts will help you tackle sort of a plan for how to do that regardless of your automation platform.
Fantastic, thank you. As a follow-up question to that, is the auto cut and run method available on the Biomech i7?
I think it would be the same answer, right? So we can help you get it set up on the Biomech.
There's also, the Beckman team. So our genomics team is actually kind of partnered with our automation team. So if there are any specific questions about automated platforms that Biomech has, so the i7 and the i5, feel free to reach out to us as well. We can kind of do a little bit of a collaboration with Andrea's team to get something viable for you on the system that you have.
Yep, totally agree.
Great, thank you. Our next question asks how size selective or number of BPS is SPRI bead cleanup versus column-based approach?
Yeah, good question. I'll let Lori maybe chime in if she wants, but I will just say the thing that I really like about using SPRI Select, so we looked really rigorously at the sizes that we were retaining at the end of cut and run, doing head-to-head of spin columns versus SPRI. And the sizes that you get are, we've tuned in the ratio of SPRI to DNA to get the same sizes. But the nice thing is that it's tunable on your end, right? So if you are profiling a transcription factor that might have a smaller footprint compared to a histone, which is, a defined size of about 150 base pairs, you can add a little more SPRI beads to tune your assay to chase those smaller fragments for transcription factor footprinting. So it's very tunable, and you can get extra beads from Beckman to do that.
Great, thank you. Our next question here says, what is the spread of off-target versus low yield, and how does low yield lead to the wrong conclusion? Presumably you can just sequence more deeply.
Not really, actually. So if you don't have any yield, it's very, very hard to get good sequencing data. You're working at the level of the noise of the assay. So truly the best indicators of success in cut and run are having good DNA yield at the targets that you're chasing. In terms of the breakdown between specificity and efficiency or yield, so I don't know the numbers offhand for the total, all the antibodies we tested, but we did look at that for those top-sided antibodies, right? The antibodies that everyone in the field uses. Interestingly, I think 90% of them were very efficient. They recovered great yield. They give you nice-looking peaks. The problem is they were not specific. So those peaks that you're seeing, you can't attribute those to the target that you think that you're profiling. So if you're using those widely cited antibodies, which vast majority of people are, that's going to be your problem. You'll get great yield, you get great looking peaks, but it's not necessarily the mark that you think you're studying.
Okay. We have another question here that says, as to native nuclei versus fixed nuclei issue, is it specific to some particular antibodies or targets?
Yes and yes. Yeah, so we always recommend, most targets, native is going to be your best option. It gives you the best yield. It's the most robust. However, if you're working with labile targets like lysine acetyl marks that are rapidly turned over by HDACs, or if you're looking at studying a chromatin protein that anchors to the chromatin by binding those acetyl marks, right? So it depends on acetylation just to bind. In those cases, or if you're having just a hard time in general, we recommend two things. One is just screen for other antibodies. Maybe the antibody that you're using is not that great. So source 4 or 5 antibodies and test them. And then the second thing would be test native versus lightly fixed. And the key is lightly fixed. If you overfix, like in CHIP, you're going to completely destroy your chromatin and destroy your yields. So just a light fix, and we have protocols for doing that in our kit manuals.
Great, thank you. And it looks like that is all of the questions that we have time for today. Thank you again, Andrea and Lori, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. I do want to remind everyone that there will be a survey that pops up at the end of this webinar. If you could please take a moment to submit your responses before you leave, we would greatly appreciate it. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.